Welcome to the Theoretical Neuroscience Podcast. I am the host, Gaute Einvoll. I just listened to an 8-hour podcast about Neuralink on the Lex Friedman podcast. Neuralink, if you don't know it, is a company founded by Elon Musk a few years ago to sort of facilitate brain-computer interfaces. And the Neuralink has really reported some impressive progress on helping paraplegic people, that is, people who can't move their limbs, to live better and more independent lives. It's based on implantation of numerous electrodes into the cortex, and these electrodes then records electric signals from neurons around the electrons. And for the Neuralink device looks a bit like a jellyfish with a circular head the size of a coin, like an American quarter, with dangling electrode arms hanging from it. And the key thing here is that after the coin has been fit into a hole in the skull and the electrodes placed in the cortex, the surgeon can cover the electrode with the skin flap that was cut open and sort of just sew this whole thing up, so there's no wire sticking out, because both the communication from and the power charging and the communication from the device and the power charging are done wirelessly. And this, of course, makes people with the device much more independent than if there was sort of like a wire sticking out that had to be connected to devices and so on. And the patient one, the first with the implant, he's a paraplegic, was interviewed and seemed very happy with the device and how it had improved his life. And a few weeks ago, I read about Alex, which is patient number 2, who actually played a video game called Counter-Strike 2, which I don't know too much about. But anyway, he played it using signals from the implant to control the mouse. And interestingly, he combined the use of the Neuralink implant with the use of the mouse control, with a mouse control device he had used before, and the combined use allowed him to move and shoot at the same time. Neuralink is, well, clearly the company that is is most well known for this kind of activity. But it's certainly not the first company who's been working on this. This goes way back. Actually, I think the first there was a company called BlackRock, or is a company called BlackRock, that made something called a Utah array. I think the patent was from 1989 or something. So that was like a similar idea, and it measured electric brain signals from neurons in the cortex. But then, it was more like a different structure. It was sort of like a needle pad of like stiff electrodes that was inserted into the cortex. And these Utah arrays have been used for many things, also to have been given some impressive results on sort of giving, for example, blind patients some kind of primitive vision that has helped to sort of help them to navigate. For example, so far I only see movies from when they navigate in hospital corridors, but nevertheless, this is sort of a good, interesting proofs of principle. Actually, our group in Norway was involved in a DARPA project a few years ago, working along similar lines with electrodes placed on the cortical surface, so not sticking into the cortex. And that was also sort of like the need of a wireless communication and charging of the device. And it was based on sort of the same kind of what's called CMOS technology that is used to make computer chips and stuff like that. But I think sort of having Neuralink joining in on this endeavor with the sort of resources and entrepreneurial skills and drive of Elon Musk is certainly a boost for the field, and particularly for the patients who can benefit from these devices. So this podcast is going to be about electrical signals, the ones that are sort of picked up by, for example, the Neuralink device, And the measurement of electrical signals have been sort of, I would say, like the workhorse of neurophysiology for the last maybe 150 years almost. So now, of course, we have also optical techniques, which are important, like 2 calcium optical imaging and so on. But they are sort of they are much more difficult to use in humans, if possible at all. Anyway, the start of I would say electrophysiology actually was a guy called Richard Caton, and he in 1875 he demonstrated in England what we now know as ECoG. So that's a electrode and was put on a cortical surface. So ECoG stands for electrocorticography. And he did it on some kind of animal. I don't remember, it was a dog or something, and was able to pick up electrical signals from the dog cortex. And then on the skull, and there was this guy Hans Berger who first demonstrated this in 1929, and this is sort of called EEG, electroencephalography, and this sort of soon became sort of a important tool both for sort of like clinical use to diagnose diseases and so on, and also for basic research. Because the limitations, or there were limitations at the time because of the electric recording technology. After all, electricity wasn't really something we controlled until like 150 years ago or so. But anyway, improvements in electric recording technology sort of has made it so that, for example, now recording inside the brains has been standard. I mean, since the 1930s at least. So there you put in sharp electrodes in the brains of animals, maybe particularly in the cortex, and then you can pick up things like spikes, which is the extracellular signature of action potentials. So they are contained in a high frequency part, while also there's also something called the local field potential, which is essentially it is the same recording. It's just that you extract the low frequency part of the signal. So this has been done since the 1930s, and of course many important discoveries were made by these. For example, and like Hubel and Wiesel's discovery of these cells in the primary visual cortex of cat that responded to bars and bars moving in particular directions and with particular orientations and so on. So these are all what's called extracellular recordings. So they are between two electrodes in extracellular space. So, I mean, you don't penetrate the cell. You have one electrode maybe just outside a cell in the extracellular space, sort of the space between cells. And then you measure the potential difference to compared to a reference electrode far away, maybe in the neck or some distant place. So it's so that's important. That's an important distinction is that it's not between an electrode inside the cell and an electrode outside the cell as when you measure membrane potentials, like one typically, or at least now it's standard to do that in when you do sort of recordings in dishes, in vitro as it's called, because it's quite difficult to measure the membrane potential in living animals. It's just difficult to penetrate single cells with an electrode and keep the electrode there, sort of with patch electrodes and stuff. An important thing about extracellular potential is that they are typically much smaller. I mean, they're much smaller than the membrane potential. So the extracellular potential are typically much less than 1 millivolt, while the membrane potential is sort of like whatever 60, 70 millivolt at rest for neurons. So it's the extracellular potential is roughly a factor 100 or more smaller than the membrane potential. Yeah, and typically, I mean, these are now, you can now measure these extracellular potentials at many positions at the same time. So we have all these sort of like new new probes like neuropixel probes, which can then maybe like in living animals measure the potentials at hundreds or even up to thousands of positions in the brain. And if you use more than one neuropixel probe, you can now actually just you can get so almost arbitrarily many neurons, at least from sort of like a few thousand, and then you end up with a lot of data. And typically, what's been considered so far, it's typically the spikes. Actually, back in the days, uh, one only one only stored sort of the high frequency part of the signals, and probably the hard drives were sort of expensive and not that sort of didn't have too much capacity. So then the low frequency part, what we now call the LFP, is was just discarded on the fly. Now we can store all these signals, and nevertheless, most sort of like scientists focus on the spikes, the higher the signatures of action potentials, and their analysis is typical statistical. So then we start with data, a bunch of spikes from recorded from many different neurons, and then you look for sort of different kinds of correlations between like the data and maybe sensory stimuli, like the visual stimuli, or maybe behavior of the animal, where the animal is, for example, as in place cells or grid cells. And so that, yeah, so that's one typical, well, that is the typical application. There's also some work on the low frequency part, where well, it was called like where the goal has often been to do what is called inverse modeling. I mean, forward modeling, that is sort of going from neural activity, a model for the neuron, and compute things maybe for the extracellular potential, for example. That's what we're going to talk about here in this podcast. But inverse modeling is the opposite. Then you start with the data, and then you sort of want to estimate where this signal, the sources that put up this signal. So one sort of typical way, well, one very sort of one type of analysis which has a lot of history is to sort of to use EEG data to estimate sort of so-called current dipoles of neuron or current dipoles, which stems from neural activity in the underlying cortex. So the current dipoles are easier to interpret. It has more like a clear interpretation. At least it's reflects currents going in and out of neurons in particular positions in the cortex. It's actually quite hard to do these kind of estimates because the problem is what we call ild post. I mean, for any kind of if you measure an EEG signal, you can in principle you can sort of explain these signals by putting current dipoles on any kind of surface inside the head. And so you can easily end up with the current dipoles placed in sort of the wrong positions compared to what is actually happening. Another application is sort of taking the local field potentials from recording sort of with an electrode that penetrates the cortex, so-called laminar electrodes, and then you can do something called you can estimate what's called the current source density, which is then a measure of where currents go in and out of neural tissue, sort of what layers and so on. So it gives more insight into sorry, insight into the functioning of the say cortex you want to understand. So these are all sort of inverse modeling. Examples of inverse modeling. You start with the data, and then you estimate sources for these signals. And another way to model extracellular signals is actually to start with the neurons rather than the data. So then the modeling is you do it's like a forward modeling of signals based on models of neural activity. So say you start with a a model of a neuron, a multi-compartmental neuron model, or it could be any kind of neuron model. And from there, you want to sort of predict the extracellular signals. So that's called forward modeling, and it's sort of I sometimes call it physics type modeling, because this is sort of kind of modeling that physics has. It's like the hallmark of physics. And over the last 20 years or so, our research group in Norway has worked on this type of modeling, meaning that the forward modeling of electric and magnetic brain signals, actually. And this summer, we, that is sort of Geir Hallnes, Torbjørn Næss, Solveig Næss, Espen Hagen, Claes Pettersen, and myself published a book, Electric Brain Signals. And it goes through sort of the foundations and applications of biophysical modeling of electric brain signals, such as spikes, that's extracellular signature of action potential, LFP, and the local field potential, ECoG and EEG, and also MEG, like the magnetic signal recorded outside the head. It's not an electric signal, but the measurement physics, sort of forward modeling, is quite similar. And so the modeling of electric brain signals is the topic of the present podcast. I think it's a quite important topic. Obviously, we have worked on it for sort of 20 years or so, and so I'm a little bit biased, but hopefully you will find it important and interesting too. So the normal format for this theoretical neuroscience podcast is that I interview an expert, but here I think our group actually counts as experts. I'm sort of considering to have my 5 co-authors on as guests, but I found out that it would be easier to instead make a solo episode where I will do all the talking. So this will be then a solo episode where I will just, if not interview myself, at least I will sort of try to, tell the story myself. And as always, I just want to say that as for all episodes, information about the episode and relevant links can be found at the webpage theoreticalneuroscience.no. And if you want to support the podcast, go to patreon.com/theoretical neuroscience podcast and choose a membership of your liking, for example, at the cost of $1 per episode. And as a member, you will get access to a video version of the podcast recordings, as well as transcripts of each episode. And note that there is a 7-day free trial period when you sign up, and I will be most grateful if you become a Patreon supporter to help me fund the production of the podcast, even if you're not too interested in looking at the videos. But I also appreciate all the other listeners, and I hope that all of you can help spread the word about the podcast to other people in the community on social media or otherwise. So, now back to the topic of today's podcast. So this is about sort of like the forward modeling of electric brain signals, starting with activities in like models for neural activity. It gives sort of like what's called like multi-compartmental modeling, biophysically detailed modeling of neurons. That is often the starting point. And the most common way for doing sort of this physics type or forward modeling of electric brain signals is to do the calculation in 2 steps. So the first step is sort of to calculate the transmembrane currents from biophysics-based neuron models, which is sort of like the things which are covered in typical computational neuroscience courses. The prime example is sort of the axon potential generation and propagation with the Hodgkin-Huxley model. But now there are also sort of like obviously many other neuron models that has been made and are used. And then the second step is sort of to use these calculated transmembrane currents to calculate electric brain signals using something called volume conductor theory. And the volume conductor theory is much less less well known, I think, in the community, even though it's actually it's a well established theory that was, I guess it's 50 years old, and it has been used in other part of, I would say, like biophysics it hasn't been that much emphasized in computational neuroscience. So now to the first step, so the computation of the transmembrane currents, and it's I mean the this kind of modeling to get the transmembrane currents. This is sort of something you like a side product of normal like traditional modeling of like the membrane potential, the dynamics, and so on in neurons. And the major event in the development of the methods for doing this is sort of the work of Hodgkin Huxley in the early 1950s, and where they developed a model for action potential generation and propagation in these squid giant axons. And the salient variable in this kind of modeling is the membrane potential, which is the difference of potentials or potential between inside and outside the cell. And the modeling approach is based on Kirchhoff's current law, which for those of us who have studied electric circuits in school, maybe, it's just what it says essentially that just the currents electric currents do not vanish. So so in this in the context of neurons, the Kirchhoff's current law sort of are used where and used to keep track of like the membrane, the currents crossing the membrane, what we call transmembrane currents, and axial currents inside the neuron, for example, along the inside of a dendrite. And the key insight and contribution from Hodgkin and Huxley was that they were able to model the dynamics of currents going through sodium and potassium channels in the membrane in terms of this new concept of gating variables. And I think this was a very impressive and important achievement. And because they were able then to fit all these parameters for these sodium and potassium channels based on one type of experiments with called a space clamp and current clamp and all kinds of things, voltage clamp. And afterwards, they sort of, when they have tuned the model, they were able to sort of then to test the model on a new type of experiment, actually, to initiate a start to initiate an action potential in one end of an axon, and then see how it spread to the other end. And the agreement in terms of velocity was about 20 meters per second, I think. Between the agreement between the model and the model predictions and the experiments were just amazing. I think it was within 10 or 20%. And it was like the shapes and of the action potential and everything looked very similar. So this was sort of, maybe that's one of the coolest and most impressive achievements in computational biology altogether. Then, based on this, was Wilfred Rao. He was this pioneer and who sort of further developed sort of this kind of theory to model not only axons but also dendrites. And he developed, used something called cable theory. And so I would say, like, by the 1970s, we had like a solid framework for biophysical modeling of neurons and networks of neurons. And today, the modeling of how action potentials, for example, are generated in neurons based on synaptic inputs, and how networks of such neurons process information, or at least send signals to each other, is it's like a key activity, key and sort of a popular activity in computational neuroscience. And this multi-compartmental modeling, that is when you sort of have neuron models with more than one compartment, like in integrated fire models, in the typical integrated fire models, then it's sort of like a standard topic in computational neuroscience courses. And we have standard tools for doing and for facilitate this kind of computations based on based on neurons with sort of arbitrary complicated geometries, meaning dendrites with all kinds of branching and complications. This can be sort of simulated with tools like Neuron. And this we had podcast, the Theoretical Neuroscience podcast number 15, was with Michael Hines, and who did this kind of, he's been developing this very, very important tool, I think, since the late 70s. So, to compare these model predictions with experiments, the typical, what comes out of these multi-compartmental models directly is the membrane potential. So if you have experiments of the membrane potential, measuring the membrane potential, then you can sort of compare your model predictions or fit your model to these experiments. And this is why, and this has been done now quite routinely, and now you have many models that have been, many neural models available for modelers, which have been made by this approach, essentially measuring the membrane potential at one or sometimes two positions inside the neuron, and then sort of make models sort of that can reproduce what you see in these experiments. But they are all done in vitro, meaning in dishes, where you can sort of have control with the patch electrodes and electrodes and everything. They're very difficult to do in vivo, that is in living animals. So if you want to compare your models with sort of measurements done in living animals, then it's not so easy to. Membrane potential predictions from a model is not sort of a little bit limited. So what you have in living animals are electrical recordings from the outside the cells, typically sort of, and then from this, the high frequency part, you can sort of these signals, you can sort of extract these spikes, which is not, it doesn't tell you about the shape of the action potential, like detailed shape on the millisecond scale, but it tells you when an action potential has occurred in a neuron in the vicinity of the electric contact. So how do we then? So that is sort of a good proxy. But if you then want to predict sort of like the extracellular potentials in detail, for example, the detailed shape of the extracellular of the extracellular potential related to an action potential, then for one thing, these multi-compartmental models are not set up to do that, because they typically assume a constant extracellular potential on the outside of the cell. So if you assume a constant extracellular potential outside the cell, of course, then you have lost the possibility to predict sort of these extracellular potentials, what they look like, and so on. So you need something extra. You need an extra modeling step, sort of linking outputs from multi-compartment neural models to these extracellular potentials. And here, this is where this volume conductor theory comes in. And this in this theory, it describes the extracellular medium as an electric conductor. So it's different from an electric insulator or like a dielectric or anything. This is electric conductor is actually a material that can conduct strum. That's Norwegian. Conduct currents. Sorry. So, and the key thing with this, the volume conductor theory is that assume that is the electric current is proportional to the electric field, or the difference in electric potentials. And this is, for those of you who have sort of studied a little bit electric circuits, this is the same as current going through this typical electric resistances in electric circuits, because then there's typically then the potential difference over an resistance is proportional to the current going through it. So that's the assumption with volume conductors, and like the used for the extracellular medium. So that's why we say that media that like volume conductors sort of our media that has this. It's also called the ohmic medium, and so in some sense it could be called a volume resistor theory also, but it's now volume conductor is what is used. So the extracellular potentials, they actually they emerge from, or they stem from currents that is sort of that comes out from the membrane into the extracellular medium. And the way to think about it is that there is this current sort of being, you can think of it as a current source that is emerges from the neuron membrane and is then pushed through the ohmic extracellular medium. And according to Ohm's law from electric circuit theory, which is applicable here, there will be a voltage change proportional to the in the medium, proportional to the resistance of the medium and the magnitude of the current emerging from the membrane. So that's sort of it's not that's all it is. Of course, this voltage change in the extracellular medium, actually, typically you get, it's like a total change from all imposed membrane currents from all neurons and all parts of the neuron. That is what is measured when we measure extracellular potentials. So in a wire, the like in electric circuits, the voltage change is only one direction. I mean, the direction of the wire. So the wire is a one-dimensional conductor of current, while the extracellular space is three-dimensional. So that's why it's called a volume conductor rather than, say, a line conductor. So that's why it's a volume conductor. Currents can go in all directions in the extracellular space. What you also, when we sort of look into the physics of this, if you have a single current injected into the extracellular medium, the extracellular space, this could be, for example, with a sharp, say, hollow liquid electrode. Then if you inject this current, the voltage around the electrode from this current actually falls low, like 1 over r. That's meaning 1 over the distance from the injected current. So this follows directly from the biophysics and the biophysics of volume conductors. And interestingly, mathematically speaking, it's the same when you do the math. It's the same formula for as the Coulomb potential, or the potential around a charge in vacuum or in a dielectric. This is called the Coulomb potential. It also goes like 1 over r. And this is often a source of confusion, because mathematically the decay of potential around a current injected into a conductor, 3D conductor, like the brain, like the extracellular space, is the same as the decay of the potential around a charge in vacuum or in a dielectric, so which is an insulator. So but the physics is, so it's the same mathematically, but the physics is very different. For one thing, the charge in the Coulomb's law is replaced by a current, or a current source, and the electrical permittivity, this epsilon thing in the denominator of the formula, is replaced by the electrical conductivity of the extracellular space. So the physics is very different, but the mathematics looks the same. So when applied to sort of computing electric brain signals from neurons, the sources of the currents, not electrodes currents, it's sort of transmembrane currents. And more precisely, it's the currents leaving from the immediate extracellular side of the membrane into the extracellular solution. Because very close, essentially at the membrane, just immediately on the outside and the inside, we have this very thin layer of charges that sets up the membrane potential. It's sort of like a capacitor charges, which is sort of just very, very close, typically within a nanometer of the membrane. So the transmembrane current that is actually affecting setting up the potential drop in the extracellular medium and gives the extracellular potentials, they are they actually also include sort of they are some sense from the currents leaving from the just outside this thin layer of charges. It's called the bilayer. Not important, but anyway. So the take home message and the important conclusion is that it's a sum both of the ionic currents that goes through the membrane, like Lie currents, whatever is sodium, potassium, but also the capacitive currents, the currents that has to do with the membrane potential changing. So both of these terms has to be included in this as transmembrane currents when you go on to do the next in the next step in the calculations. One important thing is that there is no extracellular potential generated from a single compartment neuron. Because the transmembrane current, when you include both ionic currents and capacitive currents, is always exactly zero. So integrating fire neurons do not produce, which are typically single compartment, do not produce an extracellular potential. So actually, if you want to compute electric brain signals from networks based on those, you have to do some trick. And we have in our group, I worked on that, as well as how to compute electric brain signals from firing rate models, which sort of doesn't really produce an extracellular potential by itself either, by the way it's modeled. So there are tricks to do that, so that you can compute maybe like EEG signals from neural mass and neural field models, for example, or LFPs from population rate models in cortical simulations. But we will not go into that here. So fundamentally, it's you need a single compartment neuron cannot be used to compute extracellular potentials. That is while obviously if you're just interested in the dynamics in the sort of like how spikes are generated in networks, then you can use a single compartment neurons. There are some approximations involved, but at least it's sort of there are models that produces action potentials and spikes. But if you want to compute extracellular potentials, you need at least two compartments. So what we do in general is that, we need multi-compartment models. So that is sort of models where the neuron is split into several compartments, and the membrane potential of each compartment is a dynamical variable. Because here, then the transmembrane currents in each compartment will be different in general, and they are spatially displaced, so that and they will be different and non-zero. And they will be spatially displaced, and so that an extracellular potential is generated. And the simplest neuron model giving an extracellular potential is the two-compartment neuron model. I guess it's like some model called a Pinsky-Rinzel model that was developed, I think, in the 80s, 90s, which has two compartments, one compartment representing the soma, one compartment representing the dendrite. And for this model, the extracellular potential is found by summing two contributions of the sort of 1/r type, 1/r distance type, from that you get from single sources of currents. But you have two contributions. You get one contribution from the transmembrane current in the soma compartment, and one from the transmembrane current in the dendritic compartment. And it's one more constraint, or one more important thing that follows from the cable equation, is that if you have a neuron model like the two compartment model, the sum of the transmembrane currents across all compartments of the neuron is exactly zero. So in the case of a two compartment model, if currents goes into the dendritic compartment, this could be, for example, from an excitatory input, the current sink. This this current that goes into the dendritic compartment corresponds to the current sink, because from the point of view of the extracellular space, there's current vanishing into the neuron, into the void. In some sense, from the extracellular point of view, it just vanishes. But it follows from the cable equation that even that the same current, if something goes in the dendritic compartment, the same current has to leave at the same compartment, at the soma compartment. So this will be a current source. So this pair of a current sink and equally sized current source constitutes a current dipole. So another way to state this is that there's sort of like no current monopoles. I mean, you never have if you have current injected from an electrode, like a hollow electrode, a liquid electrode, then that sort of looks like a current monopole. But the currents, when the neurons are the sources, then they always come in pairs in some sense. There's always sinks and sources, and they all exactly balance at each time point. So, for more complicated multi-compartmental models with more than 2 compartments, there's the same principle, meaning that the sum of all currents, transmembrane currents, is at all time 0. But there are obviously more contributions to this to be summed. I mean, with n compartments, you essentially have n terms in the sum, but they always then sum to 0. So, for those of you who are sort of used to sort of multipole expansions, which are often used in, for example, they use many places, also in electrostatics and electromagnetism and in many parts of physics. So we would say that in a multipole expansion, the lowest term, the current monopole, is always 0. So the lowest non-zero term is the current dipole. That is this thing that actually is helpful if you want to sort of later when we want to sort of talking about how to compute EEG signals. So because then only the lowest order term in this expansion will contribute to the signal, because higher order terms like quadrupoles and so on, they decay faster with distance, and the EEG electrodes are so far away from the neurons, typically more than a centimeter in humans, so they don't contribute. So then all we need are the current dipole moments of neural activity, or the current dipole moments of the transmembrane currents. And a huge simplification when we do this modeling of the measurements, the electrical measurements, is that the physics is linear. And this is not the case for example, processing of synaptic inputs in the neurons to make an action potential, because there you have this threshold and you need so you have this very non-linear process that the soma has in order to sort of to decide whether to generate an action potential or not. So that's very non-linear, and that what sort of also makes it hard to make good neural network models, because it's much harder to model non-linear systems. But for the measurement physics, the link between the transmembrane currents and these electric brain signals is linear. What that means is that you if you know how to compute the extracellular signal, electric brain signal, say the local field potentials from one neuron, and then you can just you can easily generalize that to a bunch of neurons, a population of neurons, because the contributions simply add up. So this is really a blessing from the creator, that this part of computational neuroscience is quite easy, in a sense that signals just add up, and that applies for all of these electric and magnetic signals. If you know how to if you know how to sort of say compute this contribution to the EEG signal from a single neuron, then you can just do this for all neurons and add up the results. So this what about this one thing we haven't talked about yet, which is a key element of this volume conductor theory, and that's how are sort of electrical signals like starting with the transmembrane currents from the neurons, conducted in this extracellular medium. For one thing, in order to conduct anything at all, it needs the liquid must contain ions. And fortunately, this extracellular medium, something called this, it's called a cerebrospinal fluid, CSF, has free ions. So it's called and liquids that has these free ions floating around is called an electrolytes. And for the CSF, it's mainly like sodium and chloride are the two main sort of ions that sort of conducts electricity. And also another point is that there's always equal amounts of positive and negative charges in the extracellular medium or anywhere in the liquid. So electroneutrality is always maintained, except exactly at the bilayers, which are 1 nanometer thick at the cellular membranes. Anyway, so the thing that the CSF is an electrolyte means that it can conduct currents. That is, so that's why it's called a conductor. And so this is very different from like the dielectrics that one learns about in introductory electromagnetism courses, which are insulators. So, but what is maybe surprising is that you can describe the conductive or equally the resistive properties, because resistance is essentially 1 over the conductivity. But the thing that we can describe the conductive or resistive properties of the extracellular by a single number, the extracellular conductivity. So, and how can this describe electric transport in this very convoluted extracellular space where there's like at least at the micrometer scale, it's like a spaghetti of axons and dendrites and pieces of glia cells and extracellular space and what have you. So how can sort of this electric transport across this sort of like spaghetti forest actually be described by a single number? And the key, well, it can, and the reason we know this is that you can test this separately. If you have 2 electrodes inserted into the extracellular space, you can check whether it's sort of like is a linear relationship between injected current between the 2 electrodes and the measured potential change between the same 2 electrodes. And it turns out that it's very much linear. It doesn't depend on the size of the current. It doesn't really depend on the frequency much either. And the reason is that, at least the way I think about it, is that these ions with the ones that conduct electricity in the extracellular space, don't easily pass through membranes. That's why we have ion channels, so that we can actually, it's essentially insulating. My membranes do not allow ions through, so that's why, except when there are ion channels there. So these ions in the extracellular space, when they are sort of get an electric field and starts moving towards in one particular direction, it has to meander between the cells. So it sort of goes in this labyrinth between the cells and is able to, able sort of to conduct current. But the key thing here is that if you sort of look at the proportionality constant, the conductivity is sort of reduced compared to the case with the extracellular liquid, the CSF alone. So I mean, the all these membranes and all cells that is in the way, that reduces the extracellular conductivity, and the reduction compared to if they were not there, it sort of corresponds to what you expect from just considering the volume fraction of the extracellular space. So it's about 20% of the extracellular space is sort of just sort of like that's where you keep that's where the liquids are, and also the reduction of the conductivity compared to if there were sort of known it was no cells there is about a factor 5. So the well, to put it sort of other words, I mean the conductivity of the extracellular medium is about 20% of the conductivity you would expect if there was only liquid there, and that is sort of corresponds roughly to the volume fraction of the extracellular of the room where the liquids move. So that is sort of like a qualitative thing. There are also some currents passing through membranes on the way, but anyway, it has been tested experimentally, so I think it's a good sort of good approximation, even though it may have some limitations when you get to the smallest scales. So the simplest formula is for the case where it's an infinite homogeneous medium, so that the conductivity is the same everywhere. So that's, I mean, of course, the whole universe is not the conductor, and then certainly doesn't have the same conductivity. But inside, say, cortex, and if you measure sort of for example, the LFP generated by a neuron, the area around the electrode and the neuron has a very similar conductivity, so it's a okay approximation to use this infinite homogeneous medium formula. There's another possible complication that is anisotropy, meaning that it's, even though if it's the same conductivity everywhere, it could still be anisotropic, meaning that it's easier to go for the ions to move in one direction compared to other directions. And and this is actually the case. I think for hippocampus, it's had been found that is like twice the connectivity in the vertical directions along this long apical, whatever it is, apical dendrites that are sort of like in a depth direction along the apical dendrites of these large pyramidal cells. It's easier to move in that direction than sort of like perpendicular to them. Sort of we have to maybe bump into all these stalks and whatever. So that's a factor 2. However, this formula can for the infinite or for this isotropy formula. Well, the formula for the isotropic case in an infinite homogeneous medium can easily be generalized to the case where there's anisotropy, so that it's easier to go in one. The conductivity is higher in one direction than the other. Actually, inside the cortex, it turns out that it seems to be quite isotropic, as inside the cortex rather than inside the hippocampus. And so this has been measured. And in any case, when we sort of, it's a bit difficult to measure, but when we sort of explore sort of effects of these sort of anisotropies, the small anisotropies that has been discovered, turns out the effects on this generated extracellular potential is quite small. Another limitation is that if this extracellular conductivity actually vary with position. And one example of this is at the border between, say, at the bottom of the gray of the cortex, layer 6, there's a border towards white matter. And white matter, that's where all the cables are with all these glia cells around. So it's more fatty, it's more membranes. So that has a lower conductivity. So at that interface, you get a step in conductivity, and that can actually affect sort of like the generation of extracellular potentials and how to model it if you're interested in potential close to that interface. Here you can actually use something called the method of images, which has been used in electrostatics to compute sort of potentials for charges in sort of in around sort of, say, close to metal surface or at places where there's steps in the dielectric constant. So this is a trick we can use. But if not, I mean, if it's the situation is if the situation is more complicated, we can always use sort of more comprehensive numerical techniques like something called the finite element modeling. So we never get stuck. Also, the measurement devices themselves affect connectivity. So, for example, in the multi-contact electrodes like the neuropixel probe, there are many metal contacts embedded in an insulating material in a shank. And actually, sometimes the effect of having a finite size metal contact can be dealt with like simple formulas sometimes, something called a disk electrode approximation. Not so important, but generally you have to explore these effects using finite element modeling. For example, what is the shadowing effect if you're on the sort of shadow side of a neuropixel probe with many contacts on one side, and then no contacts, or where there are neurons then on the other side of this insulating shank. But again, this can always be studied using finite element modeling. So, but of frequency filtering, this has been sort of a question that has been discussed. Is there sort of like frequencies where okay, so are there, when you sort of look at sort of how these electric signals or the ions are like propagated from the neural membrane to the electrodes, do the propagation sort of and connectivity depend on frequency? Could it be that sort of like frequencies, sort of low frequencies, actually currents at low frequencies actually it's easier for them to get to the contact compared to currents at high frequencies? And this is, I think this has obviously been studied because it's a big problem. And I think on balance, it's found that it's not a strong effect in the frequency range up to say 1,000 hertz, because that's sort of the frequency range relevant for like brain signals coming from neurons, because the typical the fastest thing, at least at the fastest electric thing happening in a neuron is the action potential, and that lasts sort of a few milliseconds. So that's a few milliseconds. That's sort of say 1 millisecond sometimes correspond to 1,000 hertz. So up to 1,000 hertz, it seems to be very small effects of this, the frequency dependence of the conductivity of the extracellular medium, and might increase with the frequency of the conductivity, probably because more current can pass through membranes at higher frequency because due to capacitive currents, which increases with frequency. So, but it's not a strong effect. And actually, if you want to include it, we have sort of explored this. There are small effects, and if you want to include it, you can sort of actually include it in the formalism. It becomes more cumbersome, though. So, but of course, at some small distance, the approximation of treating the extracellular medium as a simple continuous medium must break down. After all, as I mentioned, at the micrometer scale, the medium is a spaghetti-like mash of dendrites, axons, and other stuff. And so it must break down. But what this lower spatial limit is not clear at the moment. And it would be good to find out. And actually, I think if we, it's something we can find out by a combination of experiments and modeling, if the community decided to do so. So after having gone through this two-step process of computing electric brain signals, we will sort of turn to the different types of electric brain signals that are typically investigated and used in neuroscience. And for one thing, the tradition, if you do record extracellular potentials inside the cortex, then as I mentioned, there's sort of like it's the typical way to analyze them is to start analysis is to do a filtering, and you sort of do look at the high frequency part, maybe the part of the signal which is above sort of like 3, 4, 500 hertz or something, and because from there you can sort of get the spikes, the extracellular signatures of action potentials, and generally you get it from many neurons, right? Several neurons in the vicinity of the electrode, and what is called the multi-unit activity, because there's multiple neurons being recorded from. And the low frequency part is sort of maybe then below a few hundred hertz, and it's typically called the local field potential. I mean, the high frequency filtering isolates contributions from spikes because only action potentials contribute to the highest frequencies that you measure. The local field potential, the low frequency part, has many contributors, so that's different. I mean, it obviously has contribution from synaptic inputs and associated return currents. It also has contributions from calcium spikes, because they are, even though they are spikes, the calcium spikes are slower and have more low frequency components. And it also has contribution from action potentials, actually. So also action potentials have contributions at lower frequencies. So it's not that some people think that maybe it's that action potentials only have contribution at high frequency. That's not correct. The reason we do high frequency filtering is because in the highest frequencies, the action potentials are alone in having contributions. So when you sort of if you look at actually investigate action potentials, the extracellular spikes, they often have sort of components down to, I mean, significant sort of contributions to the power spectrum of these extracellular signals, down to like even 100 hertz or so, maybe a bit higher. So, and I think that the local field potential is sort of a little bit strange name. I'm not quite sure what is so local about it, really. It's more local than the EEG. Maybe that's sort of the reason. But of course, the question is how local is it? And then the second thing, why the term field, local field potential? Because it's true, it's an electrical field there. But we don't say membrane field potential, even though there is obviously a electric field going across the membrane that sets up the membrane potential. But the physical fields, in the way physicists talk about fields, is any quantity that has a value everywhere. So it could be like a temperature field telling about what, like the temperature at all spatial positions at all times. So in that sense, the LFP, or extracellular potential, is a field because it has sort of a value everywhere. But maybe a better name would be low frequency potential, because then it wouldn't even need to change the abbreviation LFP, which is sort of very much established. In any case, the forward modeling of all these signals are based on the same. Oh no, I forgot the name. I mean, also obviously the ECoG and EEG is sort of like signals that when you sort of start listing the spikes and LFP and so on, all the electric signals, we should also mention ECoG and EEG. In any case, forward modeling of all these signals are based on the same principle. I mean, you start with a multi-compartmental of a neuron, anywhere from 2, but not 1, 2 to thousands of compartments. These are typically sort of ready-made new models that you can sort of find on databases. Especially now in these days, there are like large libraries of neurons that you can download from the site of the Blue Brain and the Blue Brain project, and also and certainly also the Allen Institute. So they have both sort of large collections of neuron models. And then after you have these multi-compartment models of a neuron, you compute transmembrane currents from all compartments, and this is already produced when one computes the membrane potential. So it's just a question of actually fetching these numbers out from the simulation. And then you use the summation formula, assuming some kind of model for the extracellular conductivity. In the simplest case, this infinite homogeneous conductivity formula. Actually, the history of computing these extracellular potentials from neuron models goes back to Wilfred Rall in the 1960s. He used sort of only stylized neuron geometries like spheres and sticks sticking out and so on. And in the late 1990s, the group of Christoph Koch, then at Caltech, started doing these calculations with anatomically detailed neuron models that was sort of like reconstructed with neurolucida and so on. And our group in Norway started doing such calculations a few years later. So what have we learned from forward modeling of these electric brain signals? And obviously many things. We have worked on this for like 20 years or so. So we have explored many things, but I just go through a few maybe highlights or at least a few examples. And one thing when we started studying spikes, so what determines the spike amplitude from a neuron? I mean, some spikes, extracellular signature action potentials, are larger than others. Just like people, some people are just louder than others. But what determines the amplitude? What aspect of the neuron or neural morphology then determines the spike amplitude from that neuron? And it turned out, for example, that layer 5 neurons that we investigated have like layer 5 pyramidal cells. They have big spikes, but it's not because they have this long apical dendrite going up to the that spreads out in a tuft up at the upper layers. It's because it's a big neuron with many thick dendritic branches attached to the soma. So that is actually the best it's a rule of thumb proxy for sort of how big the spike will be is actually how many thick dendritic branches are attached to the soma, or sometimes the sum of the like a rough rule of thumb, some of the cross-sectional areas of the dendritic branches attached to the soma at the soma level. So that's why layer 5 pyramidal cells have big spikes. Another thing is that the spike shape obviously depends on a lot on the recording position. So you can even have inverted spikes compared to the normal spike. A normal spike that you typically see often have this characteristic shape with a sharp negative peak. It's called a sodium spike, followed by a blunter positive hump, which is called a potassium hump, because the first negative peak is due from the sodium currents going into the soma from the extracellular space, and then afterwards it's sort of the potassium current that is activated and reverses the pattern. So this gives this particular hump or particular shape with a down and up. But you can also get, if you put the electrode elsewhere, actually above the soma, for example, if there's like a pyramidal cell with a thick apical dendrite, you can even get inverted spikes with like the first spike goes up rather than down, and then the hump goes, the down rather than up. So that's a thing, and that's so this is different from the membrane potential. The membrane potential obviously is recorded in the soma only have one shape, right? This characteristic peak, followed by after hyperpolarization maybe. So another thing we have found is that the amplitude of spike falls rapidly with distance, at least when the electrode is outside the sort of like the bush of basal dendrites. So of course, if you're very close to the soma, then and you get a large action potential, but then you're also maybe you're sort of surrounded by a bunch of basal dendrites, and as long as you stay in that bush, you don't decay too much. But if you're outside this bush of basal dendrites, then it sort of decays much faster. Maybe more like eventually 1 over the square distance, and maybe even possibly also 1 over the cube of distance even further out. Also, a thing is that the spike becomes blunter with distance, less sharp. So you can see that it has lost some. There's less high frequency components than when the spike further away. Not only that it has smaller amplitude and size, it has also less high frequency components. So the high frequency components are damped sort of more quickly or decay more quickly with distance. And this has nothing to do with like some kind of filtering of the extracellular medium. It has to do with just the cable properties of the neurons, where the capacitive currents will plays a larger role for the high frequency components, and they are actually sort of and their role means actually that the contribution sort of actually we have analyzed this in detail. It's all in the book. But it has to do with that actually the return current for the highest frequency components returns closer to the soma current, which drives the whole thing when for higher frequency components than the low frequency components, which moves further away from the soma before returning, and that gives this effect that the low frequency components decays more less rapidly with distance. I mean, spikes recorded from axons are typically smaller, and also they have often this characteristic triphasic spikes, like up, down, up, which are different from the more like biphasic spikes recorded outside the soma, which is down, up. Typically, they are much this axonal spikes are spikes recorded outside axons are typically much smaller than the spikes recorded outside somas. However, around sort of like termination zones of axons, I mean, where they end, the contributions from axon can be quite large, actually. And for example, below cortical surfaces, if there are sort of a bunch of axons coming in and terminating at in layer 1, and where they both branch out and become like bifurcate, become like this branchy structure, and also terminate, then the signal from this thing can actually be quite large. And I think often actually when they do this ECoG recordings, then typically the high frequency signal you see with these ECoG recordings are, at least we have done some model studies, and that supports that this is actually due to axons, these branching and terminating axons, rather than spikes from somas of neurons towards the top of the surface, say in layer 2, 3. So that sort of illustrates that sometimes this sort of forward modeling can be very useful in sort of trying to figure out what these signals you measure really, what kind of neural processes they really stem from. Also, I mean, so far I've talked about spikes from individual neurons, but if somehow the several sort of neighboring neurons around the electrode then fire synchronously, the resulting net spike may actually be boosted and have larger amplitudes than what you see with single spikes. And sometimes this, I think, is called sort of like a population spike, which has been seen, I think, particularly in hippocampus, and has also been modeled there. And also when you have this kind of boosting from like synchronous firing, then also the net spike not only gets larger, it also gets broader, and it gives more contributions to lower frequencies, contributing to the LFP and making then the interpretation of LFP harder, because you have like competing contributions from synaptic inputs and return currents, or action potentials from synchronous spikes. So to the LFP, I mean, an action potential is quite standardized in the sense that it always starts with a sodium current going into the soma. So if you keep an electrode at a particular position, and you sort of what you pick up should be quite similar from it's quite standardized. So this is sort of regardless on what made this neuron fire an action potential in the first place. But the contribution to the LFP from a single synaptic on a neuron is more varied because it depends sensitively on the position of the synaptic input. For example, if you have an excitatory synaptic input onto, say, onto position, then it will give us current sink at this synapse position. So it will be sort of like a current going into the neuron from the extracellular space, so it will look like a current sink. And that means that it will be, say, if this sort of synapse is at an apical dendrite in a pyramidal cell, then there will be a negative sort of LFP hump around the position of the synapse. However, then this will be accompanied by a positive LFP hump around the soma, where a large part of the synaptic input currents actually returns to the extracellular space, and this will then be a current source. So that means that if you have say a layer 5 pyramidal neuron, then an excitatory synaptic input at the apical dendrite, you will not only get LFP at the position of the synapse or around the synapse, you will also get an LFP signal contribution to LFP signal around the soma, which then of course could be a millimeter or more away, depending on the size of the neuron and the position of the synapse in the apical dendrite. So that means that it's not that local. In some sense, it's because it cannot be more local than the spatial extension of a neuron. And if instead this excitatory synapse is positioned in the soma, the pattern is opposite. Then you get a positive, then you get a negative LFP hump around the soma, and a positive LFP hump around the apical dendrite. So you get the exact opposite. And then, of course, if you have both, then there will be cancellation effects. So that's the thing, right? That if you have so they don't. In some sense, the LFP signals from many neurons sum up, but there can be cancellation effects, or there will be cancellation effects if you sort of have different patterns like this. So that is one complication, that it really depends on the position of the synapse. And also for inhibitory synaptic inputs, the signs are reversed, because an inhibitory input gives a current source, not a current sink, a current source at the site of the synapse, and at the current sink at the sites of the return currents. So that means that if you sort of observe a negativity in the LFP, sort of around the neuron or a particular depth, the interpretation is ambiguous. Because, for example, for a spike, it's easier. A spike observed in, say, layer 3, we know comes from a neuron with its soma in layer 3, or at least not so far from layer 3, because the decays actually, typically the spikes that contributes are maybe within 50 micrometers or something. You can see the spikes maybe 50 micrometers or so, up to 50 micrometers or so away from the soma of the spiking neurons. So the signal decays rapidly. The spike signal decays rapidly over distances of a few tens of micrometers. So that spikes are much easier to interpret, at least where they come from, where the neuron is that fires these spikes. But an LFP observed in, say, layer 3 does not necessarily come from a neuron with its soma in layer 3, like it would be for a spike. I mean, it's not even from a synapse in layer 3, because it could be a return current from a neuron with its soma in, say, layer 5. And maybe also then this neuron receives a synaptic input in layer 5. So you see the LFP observed in layer 3 could be easily be due to synapses hitting sort of like a layer 5 neuron in the soma layer or in their soma, for example. So generally, a neuron receives many synaptic inputs, and the net LFP generated by the membrane currents in that neuron will depend on the spatial distributions as well as the type. So they get a large LFP contribution from synapses of a particular type, for example, excitatory synapses. They should be positioned unequally across the neuron, either all at a top or all at the bottom. So, and the interesting thing, if they are evenly spread out, then there's all these cancellation effects, and one hardly gets an LFP signal at all. But still, I mean, if this even the even if, I mean, in that case, if the excitatory synapses are evenly spread out over the neuron, you can still get a lot of excitation, so that the neurons in that population fires a lot of action potentials. So you can have neurons that fires a lot of action potentials and are sort of obviously active, but hardly produce an LFP signal at all. So they could sort of be like a stealth neuron. So I think logically it would actually be possible to make a functioning brain, or at least it's not apparent that you cannot make a functional brain that doesn't produce an LFP signal or an EEG signal at all. So this is that's more like a thought experiment. But the thing is that the LFP signal alone is not reliable it's not a reliable signal for cannot sort of reliably be interpreted in terms of the spiking activity, or that you can sort of since we work on this, I've often been asked this question, sort of like how sort of they want sort of rules of thumbs for the LFP signal, what it tells about the underlying neural activity, and that's not possible to give. Unlike for spikes, if you have like a large multi-unit activity recorded at the position, you can say, well, actually neurons around this electrode is firing a lot of action potentials. So spikes are easier to sort of have this qualitative explanation for. So, in living animals, the recorded LFPs typically stem from many neurons. So a natural question is, how many neurons contribute to the LFP recorded by an electrode placed in cortex? So alternatively, what is the size of the region of neurons around the recording electrode contributing to the measured LFP? And this is what I've called the spatial reach of the electrode. So what is the size of the region around your electrode that contribute to the LFP you measure? And it turns out here we did some detailed model study of this some time ago, and it found that the key factor determining what this spatial reach is how synchronous the synaptic inputs in the population of neurons onto the population of neurons around the electrode is. Because if these synaptic inputs to the population are random and uncorrelated, the spatial reach can be as small as a few hundred micrometers. But if, however, the synaptic inputs are correlated, the spatial reach can be several millimeters or more. So we think this can sort of reconcile experimental reports that has sort of found sort of like a large variation in this measured spatial reach of populations. So what about EEG? In terms of calculating EEG signals from individual neurons, or contributions to EEG signals, there is sort of good news and bad news. The bad news is that the electrical connectivities of cortex, like the gray matter, and the scalp and the skull, as well as the cerebrospinal fluid, are quite different. So volume conductor theory becomes more complicated, but it's still solvable, either numerically for anatomically detailed electric head models, or analytically, sort of assuming simplified head models like spherical head models and so on. And this, fortunately, at least from our point of view, this kind of the link between current dipole moments at particular positions and these EEG signals has been worked on for many decades in the EEG community. So this has essentially been worked out. So for us, starting with a neuron, we can just take advantage of these existing theories and tools. So that was the bad news. The good news is that we don't need to keep track of all membrane currents or all transmembrane currents, at least not individually, because the EEG electrodes, as I mentioned, are far away from the neurons generating the signal. So it suffices to know the so-called current dipole moment, which can then be computed from the distribution of transmembrane currents across the neuron. So instead of using, say, 100 transmembrane currents for like a multi-compartmental neural model with 100 compartments to compute the EEG signal, all you need is the three components of the current dipole moment, one and along the x-axis, y-axis, and z-axis. ECoG signals recorded on cortical surfaces are in some way halfway between LFPs recorded inside the brain and EEG signals on the scalp. And in some sense, it's more complicated to calculate than either, as so for one thing, one cannot necessarily take advantage of the simplification for EEG signals we can do for EEG signals, where we only use the current dipole, because we are not really that far away from the sources. So this dipole approximation is not always valid. And also in terms of LFP, you cannot sort of approximate extracellular connectivity to be constant as in LFP computations. But anyway, you can still do sort of ECoG forward modeling of ECoG signals, even if the numerical scheme is a bit more cumbersome. So we have sort of developed, as part of our work over the last 20 years, we have developed this tool LFPi, which is then obviously freely available, where you can sort of do now with the last version, can sort of compute sort of like starting with a neuron or network model, compute things like, well, all these things, spikes, LFPs, ECoG, EEG, and also MEG. So all of these things are covered in detail in our electrical brain signals book. And also should note that for all computed example figures in the book, we provide Jupyter notebook scripts for remaking of the figures. And then you can sort of, as the user can then sort of remake the figure, but also maybe using other model parameters and so on to sort of to see how these electric signals or whatever we calculate depends on the model parameters. And these sort of then the Jupyter these notebook scripts are then typically based on then our LFPi tool. So it also gives an introduction to the LFPi tool, but there are also other documentation for the LFPi tool that's available via GitHub, for example. So we hope this aspect of the book with all these Jupyter notebook scripts will help students or researchers who want to dive into this material in more detail. So this two-step scheme that we have talked about, first compute the transmembrane currents using sort of like multi-compartmental models and maybe the tool neuron, and then use in the second step, use these transmembrane currents to compute these extracellular electric signals, is an approximation. And the reason is that the extracellular potential generated by a neuron will, of course, itself affect the membrane potential of the same or neighboring neurons. And this is what's known as effective coupling. To model this effect, the intracellular and extracellular current must be modeled simultaneously, and the more cumbersome and computationally expensive modeling scheme must be used. But I mean, that's if you have computer power, and you can sort of still do that to some extent. Another thing not included in the two-step scheme above is diffusion, because this volume conductor theory and also the typical way that neural dynamics is modeled with the cable equation and with the tools like Neuron assumes that ions are only moved by electric fields. And however, the ion movements are also moved by both inside and outside neurons are also performed by diffusion. So, and the diffusion is always happens when you have different concentrations of, say, an ion in one position rather than another, instead, compared to another position. For example, if there is initially more potassium in the extracellular volume in upper layers of cortex than lower layers, there will be a diffusion of potassium towards the lower layers, sort of to equilibrate the concentration of the extracellular potassium. And diffusion actually will also give rise to LFP signals at low frequencies. I don't know what, maybe less than 10 hertz. And so even if there were no transmembrane currents, you would still measure you could still measure extracellular potentials due to this diffusion, what we call diffusion potential, which actually corresponds to what is like liquid junction potential, which people have to worry about when they use these liquid electrodes to model or to measure, to do current injections or measure potentials. And if you want to, so in order to do it really properly, you should, or at least include everything, and to study the combined effect of diffusion and electric fields, one needs to use you guessed it, electrodiffusing models. And these are more complicated to deal with, since one needs to keep track of all ion concentrations, not only currents and charges. But several numerical schemes are available. And with such schemes, one can study the slower processes in brain tissue where diffusion plays a role. For example, we, in our group, studied the putative role of glia cells in keeping the extracellular ion concentration of, a particular potassium suitably low, to ensure that neurons, so that neurons can operate as they should. Because a problem actually can give pathological states in brain tissue if the ion pumps are not able to keep up with the firing of action potentials, where there's always potassium leaving from the inside and going to the outside into the extracellular space. And in order for neurons membrane or the neurons potentials of, for example, for potassium, for a neuron to be suitable, suitable so that the neurons can operate as they should, this extracellular potassium concentration shouldn't be too high, or actually it should be low. So I think at rest state, when they are healthy, it's maybe as low as 3 millimolars. But if you when you fire an action potential, then you get more and more action potentials, you get more and more potassium in the extracellular space. And if there's no way to to pump out or to get rid of that extracellular potassium, this extracellular potassium, then the neurons can get in trouble. So then you have ion pumps that pump them back in. And also, but we studied, what we found is that glia cells actually can help to keep the potassium concentration in the extracellular medium low, or sufficiently low, by buffering, or by taking up actually potassium from the extracellular space and moving it to the interior of the glia cells, where they could shuffle the way into other areas of the other positions in the tissue. So, and magnetic brain signals, we haven't really talked about them, but in particular, MEG has been used. Magnetoencephalography has in many ways a similar origin as electric signals. Essentially, that is transmembrane currents. There are a little bit some differences, but anyway, forward modeling of such signals follows along similar lines as for electric signals. So, for completeness, we have thus also covered this in our book. So what are the applications of forward modeling of electric and magnetic brain signals? And I think a key thing is to get intuition about the neural origins of different brain signals. Until now, it's been mainly used as almost like treated as a black box signal to blindly use in statistical analysis. And I think it really and there has been sort of many sort of misconceptions in, for example, in like the sort of like the way people have talked, particularly LFPs, and they have like misconceptions about what the signal is and how it can be related to the underlying neural activity. So so that is sort of an obvious sort of I think important application. And but there's also, I think, this could be used to develop and test data analysis methods. For example, based on forward modeling, this kind of forward modeling, we developed a new method for current source data analysis, or like that something called the inverse CSD method, where essentially the forward model is built into the analysis. And we also used it to make benchmarking data for testing of spike sorting methods, because in a model, I mean, in order to test like, say, spark sorting methods, what you would ideally like is sort of dual recordings. You want to have the extracellular potential, and maybe I should tell what spike sorting method is. So the thing is, when you put an electrode down in, say, cortex, you pick up what's called multi-unit activity signals from many neurons. So what spike sorting is about is sort of to be able to sort these different spikes that you pick up into contributions from different neurons, because that's more interesting from an like understanding and analysis point of view. So so that's lots of students around the world are sitting and doing this manually, and this is the big manual component, which is hard work and takes a lot of time, and it's and I think can be frustrating at times. So if you get automatic methods for doing spike sorting, like that are validated and you can trust, that would be a big boost for the field. And I'm quite sure if Elon Musk had gotten his decide to do this, he could sort of fix this with his resources and drive. But anyway, so in order to test and develop these spike sorting methods, you need benchmarking data where you know both the ground truth, meaning when did the neurons actually fire an exponential, and at the same time have these extracellular potentials, because then you can sort of actually give the extracellular potential and provide these spike sorting algorithms with the extracellular potentials and see how well they are doing by comparing with the ground truth. But making this benchmarking data is difficult experimentally, because it's difficult. You need to do dual recordings, an intercellular recording at the same time as an extracellular recording of the same neuron. So that is quite complicated. Some people have done it, so there are some data. But I think like this forward modeling of signals in like a real, in a neural network model, where we can actually tune the network behavior, and so that you get all kinds of you can sort of impose all kinds of problems that you will see in experimental recordings, including experimentally recorded noise. So this could be sort of so we can produce a large set of benchmarking data can be produced to help sort of the development and testing of these various candidate methods. So I think this is, other parts of, obviously data science has a long tradition of using sort of like benchmarking data and having competitions where people can compete to actually compete on sort of successful analysis of this benchmarking data. I think that's a good idea. We haven't really there are examples of this also in neuroscience. For example, there's a brain score, which is used to sort of to evaluate these neural models that are AI-like neural models that are trained on categorization data. So there are examples, but I think we should do more of this. And certainly, if for the provide benchmarking data for the development of analysis methods for electric and magnetic brain signals. I think this kind of approach at this forward modeling can be very useful. And obviously, it's also help in this modeling can be help in the design of like these neuroprosthetic devices, like for example what Neuralink is doing at the moment. And then another application that we are interested in is sort of to use it to constrain network models, because the typical approach to model physical systems, the physics approach to trying to understand the world, is often you put up your you make your hypothesis in terms of like specific models and look at what the model predictions say and compare the model prediction with experiments. So, for example, if you want to understand the behavior of a the mouse primary visual cortex as a physical system, you can make a neuron model or a network model based on the the neurons that are known to be in this mouse visual system, hook them up according to what is known about the connectivity, and then stimulate the model with visual stimuli that has been of the same type as used to to test on mice where you have recordings, electrophysiological recordings. So we have actually done this in collaboration with researchers at the Allen Institute. So they have then first they made this. This was in a group of Anton Arkhipov. They made this large-scale network model of 230,000 neurons, and then in the first development of that network model, they used spike recordings from these neuropixel probes on a bunch of mice. Has been shown like a protocol of different kind of visual stimuli. This was recordings also at the Allen Institute, and compared the model predictions with these experiments. So then, when we joined forces with them, we sort of also computed the local field potentials from the same model, so that we could both sort of compare the spikes and the LFPs produced by the model with these experimental recordings. By the way, sort of Anton Arkhipov was a guest in one of the podcasts. What was it? It was sort of what was podcast number 10, actually, if you're interested in this large scale modeling. So okay, and then so I think this is an interesting thing. So the hope is maybe that with more and more, if you have a network model that can predict all these kind of different measurement modalities, spikes, LFPs, maybe in optical signals, and you're able to, and EEG, what have you, and then you're and then you're able to predict all the same things from the network model. You can maybe sort of then get closer to sort of a model, like a model for what the real mouse is doing. Because there are many questions related to that. For example, mice are also different, and then we just looked at experimental data from, I think it was like on the order of 50 mice recorded at the Allen Institute. They had quite different spikes and LFPs also. So there's like issues. What should we really try to fit? Is like the average mouse or whatever. So there are issues there. But anyway, that's one approach. And one thing I haven't talked about at all is this electric stimulation, and it's not covered in detail in our book, even though we have done some work on it. And but it's essentially the same physics. While describing electric stimulation and the generation of electric signals from neural activity. In fact, it turns out that the effect of stimulation of neurons with extracellular currents is the same as as can be mapped to the problem of computing extracellular potential set up by neural activity. This is something called linked via the reciprocity theorem. So these are very sort of like the field of modeling electric stimulation is very much related to the problem of modeling how neurons generate electric signals. Okay, so if you, now I'm approaching the end here, but if any of you is sort of considering to buy the book rather than spending your money on other cool things, it's actually you can get there's a possible to get a 20% discount if you buy the book from Cambridge University Press, the publisher. And I have on the homepage of this episode, I have sort of added this discount code you can use. And also, I mean, I was also involved in the production of the second edition of a textbook, computational neuroscience textbook, Principle of Computational Modeling in Neuroscience, together with David Sterrett, Bruce Graham, Andrew Gillis, and David Wilshaw, all up in Scotland at the University of Edinburgh and in Stirling University. It was published October last year. And the reason I got involved in this is and contribute to the second edition, even though I wasn't involved in the first edition, was that I used the first edition in my course, Computational Neuroscience at NMBU in Norway. And I really, I liked it. That's why I used it. And then they contacted me when they were going to make a second edition and said, well, we see you using our book. So do you have any suggestions for sort of what could be things to change or add to the second edition? And then I said, well, actually, I think it would be great to have some new chapters on the how to model the extracellular space, because and also the modeling of electrical, magnetic, and other measurements of brain activity. And I think these sort of belongs. Personally, I think these sort of topics belongs in a modern computational neuroscience course. But of course, that's me. I was worked on this for a few decades. But anyway, they thought it was a good idea, and then I ended up sort of joining in on this new edition, where and wrote so that we added a couple of chapters on this, but there's also other new things in the second edition. So I think it's really turned into a really cool book. But of course, there are also other cool and nice books that can be used in for textbooks in computational neuroscience courses. Okay, so that is what I plan to say. So take care and have a nice day wherever you are.